before we get started, I'd like to advise everyone that the meeting is being recorded. So if you do not wish to participate in a recorded meeting, you can disconnect at this time and watch the recording in the future. The agenda for today's meeting is displayed here. All participants are muted, and we ask that you remain muted during the presentation. If you have any questions during the presentation, you can post them in the chat, and we will address as many as possible during the question and answer session after Dr. Bosque's presentation. We will also paste a link to a SurveyMonkey feedback survey in the chat, and this voluntary anonymous survey will provide you with an opportunity to make suggestions for future topics or speakers. We're appreciative of your time to complete this. So at this time, I would like to introduce Dr. Sharon Hunter, the Associate Director for Basic and Translational Research in the Office of Research on Women's Health. Dr. Hunter is the faculty advisor for this SIG.
So thank you, Elizabeth. Thank you very much. And thank you all who are attending today. On behalf of the Office of Research on Women's Health, it's my great pleasure to welcome you today to this meeting of the Sex and Gender in Health and Disease Scientific Interest Group. The overarching purpose of this special interest group is to explore the influences of sex as a biological variable and gender as a social construct on health and disease across the lifespan. The SIG aims to promote the dissemination of research and foster interdisciplinary collaborations among NIH scientists who work on or are interested in aspects of sex-based research across the research continuum or in sex differences research relevant to health and disease. The SIG also serves as a platform for cross-disciplinary connections. Regarding our mailing list, you can join the SIG and subscribe to our mailing list from the SIG webpage, and we invite those of you who aren't yet members of the SIG to join us. And if you'd like to receive information from today's SIG by email, please put your name and address in the chat. And so with that, I'll turn it over to Dr. Elizabeth Barr again to introduce today's speaker.
Thank you. So Dr. Bosque received his bachelor's degree in biochemistry with high honors from the University of Zaragoza, Spain in 2001. During his graduate studies at the University of Zaragoza in the laboratory of Dr. Alberto Bosque, he studied the mechanisms of downregulation of human T-cell immune responses and their implication to autoimmune lymphoproliferative syndromes. After obtaining a Ph.D. in biochemistry and molecular and cellular biology with honors cum laude, Dr. Bosque joined the laboratory of Dr. Vicente Planellis at the University of Utah, first with a postdoctoral fellowship from the Spanish government, followed by a Matilda Krim postdoctoral fellowship from the Foundation for AIDS Research, AMFAR. During his postdoctoral studies, he developed one of the first primary cell models of HIV-1 latency. Dr. Bosque is now an assistant professor in the Department of Microbiology, Immunology, and Tropical Medicine at George Washington. Dr. Bosque has been involved in HIV cure research for the last 15 years, focusing on the discovery and characterization of signaling pathways that can both reactivate latent HIV-1 in resting CD4 T cells and enhance immune effector functions of NK and CD8 T cells. He has a long-standing funding record as principal investigator in HIV cure research, including a grand challenges exploration from the Bill and Melinda Gates Foundation, as well as an R21, R21R33, R56, and 2 R01s from NIAID, and several other grants as collaborator. So at this time, it's our pleasure to turn it over to Dr. Bosque.
Well, thank you for that kind introduction. Let me, again, first start sharing my screen, put it as a presentation mode. Now, I hope that you can see it, moving faces around. And I don't need.
Yes, it's perfect.
Before I start, what I wanted to do is go to laser pointer. So yeah, again, thanks for first the invite for me to present to your scientific interest group about our studies to evaluate the role of biological sex as well as sex hormones in HIV latency and cure strategies. But first, I wanted to start with the acknowledgement slide, so then I don't forget that what I do in the lab, or what we do in the lab, is a highly collaborative effort between a lot of the researchers that are involved in different stages of their career, from undergraduate students, lab technicians, postdoc, graduate students, and postdocs. And it wouldn't be from the sacrifice that they do and the hard work that they do that I wouldn't be able to first be able to get funding and actually being promoted in July to associate professor. So I want to thank them. And of course, I want to thank all the collaborators that I met, including my prior mentor, Dr. Vicente Planelles at the University of Utah, and several collaborators along the way, as well as the funding and the funding sources. So this year commemorates, or I will say it is, as you all know, that 40 years ago was the first reported cases of 5 young men that succumbed to a strange disease that was only observed in those individuals who debilitated the immune system. They died due to pneumocystis pneumonia. And later on, it was found that these were some of the first cases of HIV on the United States. If we fast forward now 40 years, we know that HIV continues to be an epidemic or a pandemic globally. So we know that as 2020, this is the most recent data from UNAIDS, approximately 38 million people are living with HIV. Last year only, 1.5 million got newly infected, and approximately 600 or 700,000 people still die from AIDS-related illnesses. From all the people that is living with HIV, approximately only 70% have actually access to the life-saving therapy of antiretroviral therapy. And since the beginning of the pandemic, more than 77 million people have been infected with HIV, and approximately 35 million have actually died of AIDS-related illnesses. In the context of women, we know that in every week, around 5,000 young women with ages between 15 to 24 actually become infected with HIV. This is also worse in some areas in the world, like sub-Saharan Africa, in which 6 in 7 new HIV infections are among adolescents aged 15 to 19 are actually girls. And young women with ages between 15 and 24 are twice as likely to be living with HIV than men. Some of the factors that contribute to that higher incidence of HIV is that a lot of that is coming from sexual violence and physical and sexual violence by either an intimate partner or by a non-partner. And women, they have a 1.5 times more likely to acquire HIV than those that don't experience such a violence. In general, women and girls account for more than 50% of all the new infections in 2020 and in the global infections. And this can even rise in some places like sub-Saharan Africa, where women and girls account for 63% of all new HIV infections. So now that we've seen the statistics and how this is still a pandemic, even in the context of COVID, that we need to take into account, let me go back into the basics. So let's start with what HIV and what cells HIV infects and what is the life cycle of HIV. So HIV is a retrovirus that it uses as a receptor, the molecule or the protein CD4, and as a co-receptor, a chemical receptor that it can be CCR5, but it can also be CXCR4. And is the presence of these two receptor and co-receptor what it gives a virus the tropism to infect specific cells in the immune system, in particular CD4 T cells as well as macrophages. Upon binding, HIV is an RNA virus that goes through a process of what is called reverse transcription. And it reverse transcribes its RNA into a DNA molecule that it gets integrated into the human genome. And once it gets integrated into the human genome, The virus follows the same processes that any other cellular gene, in which the viruses will have to be transcribed and translated in order to produce new viral particles. So it's been now since the mid-90s that we have developed several drugs or several therapy to target different steps in the life cycle, either by targeting in binding, by targeting the fusion of the membrane of the virus to the membrane of the cell, reverse transcription, integration, and protease inhibitors. And this antiretroviral therapy have had a lot of benefits in the sense that we know that antiretroviral therapy prevents the deterioration of the immune system, the development of acquired immunodeficiency syndrome, or AIDS, and the mortality due to HIV. Actually, antiretroviral therapy, also uses as a prophylactic PrEP, can prevent the acquisition of HIV. And finally, because antiretroviral therapy reduces the viremia to undetectable levels, we now know that a person who is undetectable will be unable to transmit or to sexually transmit the disease to its partner. The problem with antiretroviral therapy is that antiretroviral therapy does not eliminate the virus. It does not cure the disease. And now we know that HIV, even in the presence of highly active antiretroviral therapy, can persist, even though we cannot detect in plasma. So it's been more than 25 years, almost 25 years since the three groups, the groups of Dr. Celicano, Dr. Fauci, and Dr. Richman, discovered that if we take cells, blood cells from people living with HIV that are successfully treated with antiretroviral therapy, we can actually uncover replication competent HIV viral particles, indicating that HIV can establish what we call a latent reservoir of HIV, that in the presence of antiretroviral therapy, we are unable to see, but that is present even in individuals that have undergone treatment. So what we know now, 25 years later, is that this latent reservoir is heterogeneous. It doesn't consist of a subset of immune cells. Mostly is known to HIV because in fact CD4 T cells and macrophages, the reservoir is found on those two subsets of T cells, or T cell and those subset of immune cells, T cells and macrophages. But also that the reservoir can be found in peripheral blood, but it can be also found in lymphoid tissues as in other tissues and the central nervous system. So this is a heterogeneous reservoir of latently infected cells that persist in ART-treated individuals. There are several mechanisms that account for this for HIV persistence. So in here, I'm just showing a few of them. It's not an exhaustive list. The most well characterized is what we call a latent infected restal central memory CD4 T cells. So what we call with that is that these are cells that are infected with HIV and HIV is integrated into the genome, but these cells are not producing viral antigens or viral proteins, neither new viruses. And as such, they are undistinguishable from a cell that is not infected. And this is one of the main reservoirs of latent infected cells. However, we know that in some cells, this latent reservoir or this latent HIV may not be a full genome because during the process of reverse transcription, there is a lot of defects on the generation of the template of the DNA template, and they can lead to deletions as well as mutations. But sometimes these viruses can still produce some viral proteins. And that can lead that these cells are either making the immune system to recognize these cells that have these defective proviruses, and then are not allowing to the immune system to recognize the real latent infected cells. Besides that, we know that once the virus is integrated into the human genome, when the cell divides, the virus can also be transmitted into the daughter cells, and that there are different mechanisms that have been proposed, including homeostatic proliferation. We know that immune cells, as a way for a long-term survival, they will homeostatic proliferate in the presence of certain cytokines. And during that process, the virus will be also transmitted into the daughter cells because it will be copied as a normal cellular gene. it's been recently shown that even the cells that are activated during the antigen recognition and the recognition of an immune response can actually also divide in the absence of producing viral particles, and that can also lead to the expansion of this latent reservoir. And finally, there is mechanisms in which, because HIV integrates into specific sites or integrates into the genome, and it can integrate in places that can lead to an aberrant driving cellular proliferation. Finally, there is also, because ART in some tissues may not penetrate as well as in others, there is in some low level of replication that can happen even in the presence of effective antiretroviral therapy. So what do we know about the latent reservoir and biological sex? So there are two seminal papers by Nancy Archin and Eileen Scully that they first evaluated whether the latent reservoir, the reservoir of latent infected cells, is different between male and female in the context of the size. So there are several measurements of the size of the latent reservoir, and they look at, is that difference between male and females? And they did not find any major differences in the size of the latent reservoir. What it was found by Eileen Scully and colleagues is that although there is no differences in the size, there may be difference in the way that this HIV is transcribed, because one of the things that we know is that estrogen can block HIV transcription. And as such, estrogen can block this virus for being expressed. And this adds to the already a long list of differences that are known between biological cells in the context of HIV infection. So, but why is important this latent reservoir? And the importance of this latent reservoir comes because if an individual that is living with HIV stops taking antiretroviral therapy for any reason, the virus is going to rebound and the disease is going to progress. As such, the field is looking for ways to eliminate this latent reservoir in two kind of different ways. One, it could be that we completely eliminate this latent reservoir so the virus never rebound, or that it's modified in a way or is reduced in a way, or we boost the immune response in a way that when the virus rebound, the immune system will be able to actually control the virus and block the progression of the disease. So why those are the two major, although there are others that are being investigated, because we have precedents from that. We have what we call a sterilizing cure in two individuals in which the virus has been completely eliminated from their body, and that is the famous Berlin patient and the London patient, in which these two individuals receive a bone marrow transplant because of a cancer, and the bone marrow transplant was done using donor cells from an individual in which carries a mutation, a truncation of the CCR5 co-receptor. This is a naturally occur. This is a mutation that is observed in certain populations and is not a highly frequent mutation deletion, but these cells are now resistant to HIV infection by a virus that uses CCR5 as their co-receptor. And as such, the transplanting this new immune system that is protected by HIV led to a long-term or elimination of latent infected cells. But there is also another study, the Visconti study. In this case, these were individuals who were HIV infected, took antiretroviral therapies, and for one reason or another, they stopped taking antiretroviral therapy. And after they stopped taking retroviral therapy, some of these individuals control HIV infection. And the mechanisms by which these individuals control HIV infection are still under investigation. But it gives us the hope that we can either completely eliminate the virus, or we can promote an immune response and promote immune responses that are going to lead to functional cures. So when I talk about HIV cure, I like to divide HIV cure into whether cures are going to be highly specific or highly accessible, or whether we need to find a cure that it has a little bit of both, right? Because we need to find cure approaches that are going to be targeting specifically HIV that will lead to lower side effects. But there also we need to take into account that there is 37 million people living with HIV, and we need to be able to find treatments that are accessible for everybody. And when I talk about cure strategies, I like to do three main groups. The first will be the genetic strategies that I think are the most specific strategies, as bone marrow transplants, as the Berlin and London patient, in which a new immune system was able to promote clearance of the latent reservoir. So we can do that because they're, for doing a bone marrow transplant, you need to have some compatibility between the recipient and the donor. That the best recipient is your own cells, and now we can genetically engineer bone marrow cells that are actually resistant to infection by mimicking the deletion on the CCR5 co-receptor. And I think in the last year, the success of CRISPR-Cas9 in gene editing is even put into the front for into using this to completely remove HIV that is integrated into the human genome from the cells. The second category of cure strategies will be immunotherapy strategies, similar to those that are being developed for cancer, including CAR T cells, ex vivo expanded HIV-specific T cells, or the use of other effector innate cells such as gamma delta T cells, NK cells, and others. And lastly, it will be those pharmacologic strategies, which we know that pharmacological strategies tend to be they tend to have higher levels of side effect, but I think they may be more accessible, and it can be either it's been proposed as either the reversal of immune exhaustion that exists in HIV-infected individuals, as well as what is called a shock and kill strategies, or block and lock strategies. And for today, I'm going to talk about the shock and kill strategies that are the strategies that we are working with. So what is shock and kill strategies? And this is a simplified version of what a shock and kill strategy will work. As I said, HIV, when it's latently infected, is integrated into the human genome, and they will be expressed little to no viral proteins. And that, as such, it is completely evades the immune system because it basically looks like a normal, like a non-infected cell. So the idea with a shock and kill strategy is that first we have a shock, and the shock can become with what we call a latency reversing agent, or LRA. LRA is going to be a small molecule that will enhance the transcription and translation of HIV in a way that now the cell become a target for the immune system, either CD8 T cells, NK cells, or any other immune effector cell. This obviously has to be done in the context of antiretroviral therapy, because as we are reactivating this latent virus, these cells are going to be able to produce new viral particles. And you want that these new viral particles will not infect cells that are otherwise uninfected. So when we think about the shock and kill strategies, although this looks really a simplified model, so what we are starting to look at is that, well, we know how, or we know the basics of shock and kill strategy, and we know how it should work. And why they haven't worked, we are still trying to understand why some LRAs that have been already in clinical trials haven't, although they were successful in different model systems, they are unsuccessful to reactivate or to clear the latent reservoir in vivo. So when we are thinking about shock and kill strategies, so we have to think about the potential factors that can influence these strategies. And these are the ones that I decided to talk about, but there may be other ones, and some other people may have other ideas, but there are many things that are going to influence these shock and kill strategies. Not only biological sex, but also the presence of sex hormones. As I said, estradiol represses HIV transcription. So the presence of estradiol is going to influence the activity of an latency reversing agent. But also the genetics. Obviously, when we are looking at different individuals with different backgrounds, that can also influence how the response to a different LRA due to maybe the target that we are trying to target, or the protein that we are going to target presents different polymorphisms, and that can lead to the different effect of the activation of different signaling pathways, but as well as the immune system. Age is going to be another factor that is going to influence shock and kill strategies. because we know that age influences the immune system, and the immune system, age leads to senescence of the immune system. And as such, shock and kill strategies may not work as well with age. So I put another category here, that is the environment. And when I'm talking about environment, I'm talking about things that will influence the immune system, that are known to influence the immune system. We know that diet influences the immune system. We know that exercise influences the immune system, but also drug abuse, that also cigarette smoke. So we know that all that is going to also influence the shock and kill strategies. Antiretroviral therapy regimen, we actually don't really know much about how these small molecules, antiretroviral therapy is a small molecule, and we know that there is off-target effect of these small molecules, whether or not they are actually influencing shock and kill strategies. And finally, we know that in people living with HIV, even in the presence of antiretroviral therapy, even in the presence of the viral suppression, there is a chronic immune activation that leads to what is called an immune exhaustion. And we know that then the kill part of the shock and kill strategies are not going to work as well unless we revert that chronic immune activation. Obviously, this is too much to focus for a small lab. So what we are now focusing particularly is in whether in the context of shock and kill strategy, sex differences, sex hormones, or age play a role on that, on these cure strategies. So how do we do that? So the first thing that we need to know is that there are not actually unique markers for latently infected cells in vivo, and the low frequency are really harmless to study the latent reservoir in vivo. And as such, several of us work with model systems that can be used to study HIV latency and preclinically develop cure strategies, and can be either cancer cell lines, primary cells, or cells isolated from viremic participants, all the way to use mouse models or monkey models, with the idea that to develop strategies that can end up in human clinical trials for HIV cure strategies. But when we do that, we also have to take into account that whatever cure strategy we are trying to develop has to take into account the diversity of people living with HIV and how that diversity could influence those cure strategies. So, but I need to talk about models, because this I borrowed from one of my M.B.A. classes, and it was talking about financial models. And when it was presented to me, it was like a light bulb sign on the top of my head, because it actually, when we are working with model systems, we need to know that the most important question about any latency, any biological model, is how wrong it's likely to be, but how useful it is despite of its assumptions. Because we must start with models and then overlay them with common sense and experience. And I think my favorite one is this one, is all models are wrong, but some models are useful. And with that, when I started my postdoc, we developed a primary cell model, now more than 10 years ago, to study latency. And what we've been doing is, over the past 10 years, we've been refining this model. How are we refining this model? Is because we are comparing with the reservoir found in people living with HIV. And then we can see the similitudes between the reservoir that we established in the lab to the reservoir that is in patients, in people living with HIV. And then say, what are the similitudes, but what are the differences? And if there are differences, why? And how can we actually manipulate the model system to become closely related to that of the reservoir found in people living with HIV? On the other hand, we've used extensively this model system to develop novel latency reversing agents. So for the talk of today, what I'm going to do is talking about our studies evaluating the role or the influence of biological sex, age, and HIV status in an in vitro primary cell model of HIV latency. So as I said, it's a primary cell model. So we start with blood from any individual. It actually is not restricted. We can do it even from blood isolated from people living with HIV. So we isolate PBMCs by density centrifugation. It's a normal technique. And then we isolate naive CD4 T cells. As I said, CD4 T cells is the main reservoir, or one of the major reservoirs for HIV. So we are using these naive CD4 T cells as a starting point. Once those naive CD4 T cells are isolated, The process of generating latent infected cells takes approximately 2 weeks, 17 days. So, and it starts with the activation of the T cells, because what we wanted to do is generate and expand the number of cells that we have in culture. So we activated through the TCR that mimics a T cell activation, like the cells are going through an immune response, and we expanded in the presence of cytokines, in particularly IL-2, that allows the cells to grow. Once the cells are expanded, what we do is spinoculate. Basically, we put virus in contact with the cells, and we do a spin infection. And then we can measure if cells that are productively infected, because we can follow this by flow cytometry, in where we look at for the downregulation of CD4 by HIV. Is something that is known, that NEF and BPU and envelope can downregulate the expression of CD4 in the surface of the T cells, and then the expression of a specific HIV protein, GAG, that is part of the capsid. So as you can see here, we can detect a number of cells, in this case 6%, that are infected with HIV. At that time point, what we do is that a step that we call the crowding step, that these cells are put in 96 well-run plates for 3 more days. And we do that because that allows the cells to go into the bottom of the wells. And then HIV is easier to transmit in the cell-to-cell contact than as a free virus. That cell-to-cell contact, cell-to-cell transmission allows to increase the number of cells that are infected. So with that, and here I have 15 or 16 male and female donors, but we've done over 100 and 200 donors over all these years. So what we have is that, we have an initial infection, approximately 5%, and we don't see any difference between female and male donors. And then the virus, after the crowding step, it replicates. And again, we don't see a major difference in between male and female donors. So, but we wanted to look at whether the replication from day 10 to day 13, is there any difference between female and male? And what we observe is that in general, the virus replicates 4 times during these 3 days, and there is not a major differences between female and male. But one of the things that you can observe is that there is a lot of variation between the different donors in both the male and female. So we were trying to understand, why is that difference? And just in this case, we look at age. And one of the things that we observe is that it actually in female CD4 T cells, but not in males, there is a correlation between the ability of the virus a negative correlation between the ability of the virus to replicate and the age of the donor, indicating that cells isolated from older donors tend to replicate the virus less in this in vitro system, and we don't know why yet. So now let's go on to how do we generate latency. So now we have infection, we introduce another 4-day step in where we introduce antiretroviral drugs. So we introduce 2 or 3 different antiretroviral drugs to stop any further viral replication. And at day 17, what we have is a mixture of population of cells that are CD4 negative, some of them that are P24 positive. So we define latent infected cells as those cells that will not be producing any viral protein. And that is easily seen in that those cells will not even downregulate CD4. Based on that, we sort cells based on their CD4 expression using a magnetic beads, and then we purify this population, what we call a post-sort, and where we just can see all the infected cells are gone, and we just have these CD4 positive, P24 negative. And now we ask, is there any latent infected cell in here? And that can latent infected cells be reactivated. So we, again, put it in culture in the presence of antiretroviral therapy with different stimulations. In here, we have an unstimulated condition, and I forgot to mention that we always have an uninfected culture in parallel to help with our gating. So what we see is that if we don't add any stimuli, just basically culture in media, RPMI media, is that what we see is that there is no increase on P24. When we add what we call a weak stimuli, that it will be the gamma cytokine IL-2, we do see now that there is a proportion of cells that actually produce P24 and downregulate CD4. When we use our antigen stimulation using antibodies that recognize CD3 and CD28, that is the maximal stimulation that we can give to a CD4 T cell. And actually what we now see is that in this donor, 3.62% of the cells are able now to produce HIV. And because this is done in the presence of antiretroviral therapy, this is the novel expression of HIV viral proteins coming from the latent reservoir. So when we look at the role of H and biological sex, what we observe is that there is no differences in any of the stimuli that we use in here in the reactivation of the latent virus. There may be some a small reduction in female compared for male donors, but it was not statistically significant. And what we also observed is that the ability of the virus to reactivate, or what we call the latent infection, and the ability of the virus to replicate in culture, there is a linear correlation, and this linear correlation is more clear in male donors, in which the higher the virus is able to replicate, the more latency that is generated. So, and that we've done this now in multiple other donors, and this correlation normally persists. So when we are comparing female and male, we can look at the latency ratio, and that is basically what is the proportion of infections that become latent based on the productive infection. And again, what we observe is that there is no differences between female and male donors, but again, we observe that in some donors, regardless of biological sex. Some of them, they are really well established in latency, and some others establish a lower level of latency. However, there is no correlation on age based on that. There may be other intrinsic differences, like genetic differences, that they make be that in some individuals, the virus is more prone to establish latency than to establish a productive infection. So as I said, we work a lot with latency reversing agents, and I just want to bring this back just to bring back into the latency reversing agent area, where we are looking for ways to reactivate the latent virus. I show you that in this model, we can reactivate this latent virus based on the production of viral proteins. So, but the stimuli that I just showed, like CD3, CD28, is not druggable because you cannot use you cannot promote a strong T cell activation as it can have bad consequences. So we are looking for small molecules or small molecules that will reactivate this latent HIV. And as a field, there are many latency reversing agents that belong to multiple different classes. Either there are TLR agonists or epigenetic modifiers, and mostly all these epigenetic modifiers are drugs that are coming from the cancer field, as well as PKC agonists that are a mimic of the TCR stimulation, but there are some of them that, for example, ingenols are used in atopic dermatitis, and they are activators of transcription factors that are required for HIV transcription. So we haven't tested all of these in this model, but we've tested a few of them, and we compare the ability of these latency reversing agents to reactivate HIV. So, and we compare the activity of ingenol, that is a PKC agonist, and we didn't observe major differences in the ability of ingenol to reactivate between female and male donors, neither biostatin 1, that is another PKC agonist. So we also tested HOD-HPT, which is kind of an in-home LRA that we discovered that it targets STAT5 by modifying its function, and it reactivates HIV. And this LRA there is no differences either between female and male donors. We have also tested a TLR agonist, TLR1 and 2 agonist PAM3 CSK4, as well as the HDAC inhibitor SAHA, which has been in clinical trials. And we didn't see any major difference on steady state level of the ability of these compounds to reactivate HIV in this model system between female and male donors. So conclusion so far is that primary cells can actually be used to address the role of external factors in HIV infection latency and latency reversal, but obviously at the cellular level. So we are just looking at the cellular level. We have to think as a model that there is a lot of other things that are happening that we cannot mimic. In our studies, no major differences were found in the establishment or reactivation of latent HIV based on biological sex. And we found that age may influence the ability of HIV to replicate in CD4 T cells isolated from female, but not from male donors. But obviously, we work with models and we know its caveats. And these are some of them, is that first, we cannot reproduce how other cell types of the reservoir may behave. As I said, there are multiple cell types. There are also macrophages. There are also tissue resident cells, which the phenotype is maybe not the same as the cells that are generated in the lab, but we know that. Also, we know that the reservoir is also there is an interaction between immune cells and the reservoir, and there is selective pressures on this latent reservoir, which we cannot reproduce in vitro. However, in spite of these caveats, this model also can be used to investigate the factors that control the establishment and reactivation of the latent reservoir in CD4 T cells. Since we published that, we've continued to investigate the role of biological sex. And for a time, I'm not going to be specifically talking about each one of the publications, but I will do kind of an abstract. So in this study led by Amanda Macero in my lab, a research scientist, we evaluate different toll-like receptor agonists, and in particularly those of the TLR7 agonist category. And when we did this is because TLR7 agonists have been already in clinical trials for HIV cure, but TLR7 receptor is actually encoded in the X chromosome. And it's known that its activity is influenced by biological sex. Furthermore, there is recent studies indicating that there is a specific polymorphisms which can influence the expression of TLR7 in female but not in males. And as such, evaluating whether biological sex has a role in reactivating through these pathways will be really important if we are moving this into the clinic. Then in another study in which we led by a graduate student who just graduated and moved to industry, but she looked at whether I showed you that the virus can be reactivated, but now we know, and this was something that it was uncovered not long time ago, a few years back, that there is a portion of this virus that is intact, the whole HIV genome is integrated into the cell, but it cannot be reactivated. That is what is called an intact, non-inducible latent HIV reservoir. So based on this information found in people living with HIV, we evaluated whether in this model we are able to, or this model generates this intact, non-inducible latent HIV, and we found that yes, it is induced, and it is induced similarly between male and female donors. But now we have a model in which we can study what are the biological processes that determine that an intact provirus that is integrated into the human genome cannot be induced in an easier way than using cells isolated from people living with HIV. And finally, this is a work that it was done in collaboration with my previous mentor, Vicente Planelles, and Matt Sunaski during his PhD, and while I was still at the University of Utah. We observed that monocyte-derived macrophages from female donors, but not, that if we compare, if we compare HIV infection in monocyte-derived macrophages from male or female donors will always be more resistant to HIV infection than male macrophages. And that was because there is the activity of SAM-HD1, that is a restriction factor for HIV, was higher in female donors than in males. And the mechanisms by which that was happening was still unclear. So these are some of the studies, but what is next? What are we doing? So one of the things that I haven't talked today is what is the role of sex hormones in HIV latency reversal? Because as I said, it's known that estradiol can interfere with HIV transcription. So as such, the estradiol will influence HIV latency reversal because we need transcription for latency reversal to happen. So, but we took that and we are trying to, using this model system, to expand the studies of estradiol to first other sex hormones such as progesterone and testosterone. And this, this kind of is a new area of research for the lab. And when I was, writing up a grant on this and trying to get information about what levels of sex hormones are in male and female and what are, so I knew it, but it was not as clear that not only there is differences between male and female and the levels of sex hormones, but also that sex hormones fluctuate during different life stages. And the good thing of having a model system is that we can model these different life stages using different concentrations of the different hormones. That whether it's during gestation or during menstruation, so they change. But also, one of the things that we are really interested in is that we know that transgender females are more prone to HIV there is a higher prevalence of HIV infection. And as such, these cure strategies will also have to be used in transgender populations. And in transgender populations, we have a hormone supplement that can also influence this latency reversal or this shock and kill. And I think this allows us, using a model system, to evaluate how hormones interact with the immune cells at different levels. So not only that, we are also really interested on the kill. And what role play age, biological sex, and sex hormones in both NK and CD8 T cell cytotoxicity activity. We focus mostly on NK cells so far. We've been looking not only what role plays age and biological sex on NK function for HIV, but also for cancer. And that is something that we are actually finishing a publication upon. But also we wanted to expand it to the sex hormones, as well as to look at CD8 T cell activity. So with that, I think I'm on time for the discussion. And I just want to acknowledge again the people in the lab, because it's who really spearhead most of this work and is doing the work. As I said, it takes a village to do these studies. Again, I want to thank all my collaborators, and both at GW and elsewhere, as well as the funding sources and the funding. And I will now take any question.
Thank you very much, Dr. Bosque, for such comprehensive presentation. And now we have time for the question. As a reminder, if you have any question, please post them in the chat. And we also posted the SurveyMonkey link in the chat box to receive your anonymous feedback. And please ask your question. You can unmute yourself if you want. And we have a question for Dr. Bosque, who has a good start.
This is Sharon Hunter. Thank you so much, Dr. Bosque. This is a really wonderful, very comprehensive presentation and very detailed. I have a question that actually two questions. The first is, you talked about the reservoirs, and so do you notice if these reservoirs change with age? So you still talked about a reservoir in the brains, in cells in the brain and different areas. Do you notice a change in the reservoir with age and or sex, or the intersection of both of those?
Yeah, so there are, in vivo, from people living with HIV, there is studies. The reservoir is mostly stable over age. I don't know if it is, I would say that we know that it like the half-life of a latent infected cells is approximately 4 years, at least for the CD4 compartment. For other subsets, for example, in the brain, it is a little bit more difficult because of the accessibility to get samples on the brain at different times, unless and I think that is one of the challenges that we have, is that because these are such a small percentage of cells in any part, even in lymph nodes or in the gut, is the sampling is going to be really important, and whether or not we can do that longitudinally. So it's been done from peripheral blood because there is studies looking into that. When they look at also between male and female, that reservoir decay that is by several groups has been estimated to be kind of approximately 4 years. There is no major differences between male and female over the time using peripheral blood. But obviously in tissues, it's going to be a little bit more difficult to assess.
Sure. Okay. Thank you very much. Thank you. Okay, I guess I have another question. So in your model, as I understand it, where you're generating these latently infected CD4 cells, if I heard you correctly, you said that there are differences in replication between males and females. Okay, but I was sort of interested, and I think you answered me. Are there differences in the infectivity? So you have this peripheral blood that you're taking, and then you have to infect them. And so are there differences in the male and female donor infectivity? I think you mentioned, I think you had a publication about that. Is that relevant to this discussion?
So that was for macrophages, the other cell
Oh.
study that HIV infects, in which the studies were done using male and female donors in parallel, and will show that, and it shows that there is a lower infectivity in female donors, but that didn't happen on CD4 T cells. So no, but that is a good question. That is in that publication, it's done in parallel where we look at the CD4 T cells,
right,
infectivity, and recently isolated CD4 T cells or macrophages derived in the lab, and then that resistance or that difference in infectivity was only observed on the macrophage subset, but not on the CD4 T cell.
Okay, okay, thank you very much. And then I was thinking about the age of the donor also, whether infectivity of the donor's age influences infectivity.
We haven't seen any major, and I know that I see that there is a few people in my lab that they know more, because one of the things that we've had in some cases with older donors, we have a lower recovery of these naive immune cells, because they may have less naive CD4 T cells. When we look at the infectivity versus age, actually, I know that Rachel, who is a postdoc in my lab, she's been looking into that with some small molecules, but I don't think that we observe any difference. I don't know if I want to put her on the spotlight. I know that she's here because I see the face, but she's looking into that in the context of some small molecules that block HIV infection, and whether biological sex or age can influence the activity of these. And then we may have some data looking into the age and just the infectivity of the virus.
Certainly, thank you very much. Thank you.
Pam, did you want to unmute and ask your question?
Yeah, I thank you. This was a really interesting talk. I just want to, I'm a clinical type, so I want to have an understanding of your cell culture. You add donor cells, but you mentioned something about other cells being there. Are this all the cells in culture from the same sex, or do they differ? And perhaps would, maybe you could comment on that, and whether or not that might be an interesting strategy to look at.
Yeah, no, normally the cells are all from the same sex because it's the same donor. Like in the in vitro model system of latency, because we start from any donor, and then
so the macrophages and everything are all from the same donor.
Yeah, and then when they are comparing infectivity, they are parallel cultures, for example, and they are generated at the same time. So from the two different, a male and a female donor in that study, it was done in parallel. To take into account, because macrophages take a long time to differentiate. So that was done, they were not co-culture, male and female donors. They were all cultured separately.
Okay, and with regards to thinking about the age piece, do you know whether or not these females were menopausal or not?
Well, unfortunately, we don't have a lot of that information, because we get the blood from a blood bank, and we get their age and their biological sex, but we don't get any information because of what is their status, of the hormonal status, or whether they are in hormone replacement therapies or anything. We don't get that. Is something that I will, in the future, would like to look at into, but we don't have that information. And because we get kind of a leukoplaque that is concentrated white blood cells, we cannot even get, like, we don't get plasma or anything to actually measure. That is the caveat, because we don't really know.
Okay, because it would be easy to imagine that the people who are over age 50 would be menopausal, and it would be interesting to think about that.
Yeah, no, that's true, because with that, they may explain some of the other projects, some of the biology that we observe in, regardless, mostly on aging. So in the function of other immune cells, where we do see a clear effect of age on, for example, NK function, and that can be something that it will be interesting to look at.
Thank you.
Ron, John, did you want to unmute and ask your question, or would you like me to ask it for you?
Yes, thank you. Yeah, I was wondering, and I think it was touched upon during the talk, but I wanted to ask, how does hormone, administration of hormones for various reasons, whether, for in aging populations or people who are undergoing trans sex change, etc. How does that impact HIV infectivity or latency? And if you don't have information from HIV, is this something that has been studied with other viruses? Thank you.
Yeah, no, that is a really, really good question. And actually, we've been trying to look at the reservoir on trying to get, I had a collaborator, Dr. Mimi Ghosh, here at GW, which we were trying to look at how first in vivo that will influence the reservoir in vivo in transgender individuals that are going through hormonal replacement therapy. What I can say is that in this in vitro model, we can mimic that in a sense in that we can provide male, for example, male donor cells with higher levels of estrogen, and look at how that influences HIV infectivity and latency establishment in the model, because it will be really important to see not only, whether, if we know that estrogen in female donors already influences the ability of latency reversal agents to work, whether in a male, a transgender female will also, the hormone will have that negative effect on the activity of the LRA for cure. But those are really, regarding other viruses, I actually don't know. I don't know that, but it is a really important question to address, is how even the reservoir dynamics is in transgender individuals compared with cisgender. But I don't have information. I don't think that is being done.
Thank you. I believe probably more studies are needed then, but thank you, appreciate it.
Thank you. And I just have a comment, that we've seen really well documented the under enrollment of women in cure studies, and it's certainly probably a complex combination of factors, including the invasiveness of many of the sampling procedures, and so I think work like this is really important to identify signals, that can, or potential areas of inquiry for future studies. And, I'm interested to see how this moves forward. And thank you for your talk today. And I'd also like to encourage everyone who would like to receive information from today's talk or about the SIG. If you're not already on the SIG list, you can go ahead and put your name and email address into the chat, and we'll be sure that you get information. I'd also like you to save the date for the next Sex and Gender and Health and Disease SIG webinar, which you should be able to see here on the screen. It will be September 21st, featuring Dr. Katie Dahlke from Penn State, giving a presentation about LGBT mental health findings from the report of the National Academy's consensus study on sexual and gender diverse populations. So hopefully you can join us for that. Another upcoming ORWH event next week on Thursday is a webinar, Diverse Voices, COVID-19 and the Health of Women. And that virtual event will feature Drs. Lisa Boleg and Tonya Poti discussing applications of intersectionality in COVID-19. And last but certainly not least, ORWH has a suite of e-learning courses to further satisfy your curiosity about sex and gender and their influences on human health. And so we encourage you to take a look at the ORWH website. We have the Bench to Bedside course, the Sex as a Biological Variable Primer, and an introduction to the scientific basis of sex and gender related differences. So thank you all for attending. And Dr. Hunter, do you have any closing thoughts?
No, thank you. And thank you, Dr. Bosque. It's been a wonderful presentation, and thank you for providing information for our SIG audience.